Thank you. Thank you for inviting me. It's a pleasure to be here and to get to talk about PI3 kinase and the connection between diabetes, obesity, and cancer. I do have some conflicts. I have research contract from Pfizer, consult for GSK and Novartis, and I will be talking about a couple of Novartis PI3 kinase inhibitors. I'm also a stockholder in Agios and Bristol Myers Squibb. So the talk will have two parts to it. In the first part, I'll give an overview of PI3 kinase signaling network and how it controls metabolism and cell growth. And in the second part, I'll talk about the connection between obesity, insulin resistance, PI3 kinase, and cancer. So we discovered PI3 kinase in 1988 because of its co-purification with a variety of oncoproteins that caused cancers in chickens and mice. We found it as a phosphatidylinositol kinase activity, but to our surprise, it was phosphorylated inositol on the inositol ring that hadn't been previously observed, the 3 position of the inositol ring. And it generated a whole family of new phosphoninositides that had been missed in the previous 30 years of research on this lipid. And in particular, it produced this triply phosphorylated lipid shown at the bottom, PI345P3, which for the rest of the talk I'll just call PIP3. About 10 years after we discovered PI3 kinase, a tumor suppressor gene that had been identified by positional cloning as being lost in a large number of advanced cancers, and whose sequence was predicted to be a phosphatase, was identified by Jack Dixon as a phosphatase specific for the 3 position, the phosphate to 3 position inositol ring, indicating that this tumor suppressor gene was precisely reversing the effect that PI3 kinase was having on production of this lipid. Now, obviously, the PI3 kinase pathway did not evolve to cause cancer, and if we look through evolution, we can get some understanding of what this pathway really does, or what it intended to do as it evolved. This pathway is conserved in all multicellular organisms that have been investigated, but is not found, at least the triply phosphorylated lipid PIP3 is not found in Saccharomyces cerevisiae, for example. And so what this pathway really is designed to do is distribute nutrients into individual cells in a multicellular organism once that organism acquires food. And it does so by activating glucose uptake and other nutrient uptake and driving anabolic processes. And as long as this pathway is functioning normally and P10 is functional, then every cell in that multicellular organism only grows at the appropriate time when the appropriate growth factor is present. And the initial growth factor found in flies and worms that turn on this pathway was insulin IGF-1. Now, in humans, we have other growth factors that can also activate PI3 kinase, but still, insulin is by far the most dramatic mechanism for activating PI3 kinase in tissues that have insulin receptor. So what happens in type 2 diabetes, as we learned in great detail last night from Dr. Sample, is that there's a defect in the ability of insulin to activate PI3 kinase in downstream signaling. And as a consequence, liver, muscle, and fat tissue is impaired in its ability to mediate glucose homeostasis, and this results in hyperglycemia. And so this can happen by a variety of mechanisms, which we don't fully understand. It's, I think, in some ways rather embarrassing that as important as this disease is, and it is arguably the most rapidly growing disease in the Western world, in fact, the world in general, we still don't fully understand the various mechanisms by which this can occur. Now, in contrast, if there's a constitutive activation of PI3 kinase, typically in an epithelial cell, and this can occur by a variety of mechanisms. It could be an activating mutation in PI3 kinase, the gene encoding PI3 kinase itself. It could be loss of P10, or it could be the activation of a growth factor receptor, either overexpression or mutations in growth factor receptors that turn on PI3 kinase. In any event, these series of events can result in constitutively high levels of PIP3 in those cells, and that will drive high rates of glucose uptake, increased redox stress, more DNA mutations, and ultimately result in full-blown cancers. Now, the actual biochemical mechanism by which this occurs, you saw a little bit about last night. This little cartoon will refresh your memory that when insulin binds to its receptor, it causes autophosphorylation receptor on tyrosine residues that are in a sequence context that recruits the IRS proteins, IRS1 for example, that themselves then become tyrosine phosphorylated at sites that are optimal for binding to the SH2 domains of the regulatory subunit of PI3 kinase, P85, encoded by the PIK3R1 and 3R2 genes. P85 is tightly bound to the catalytic subunit of PI3 kinase, encoded by PIK3CA. As you'll see in a moment, there are multiple genes for PI3 kinase, but it's only the PIK3CA gene that's regulated by insulin. And that's also the one that's mutated in cancer. And it encodes a 110 kilodalton subunit that has the catalytic activity that produces PIP3. And this engagement of the SH2 domains turns up the catalytic activity, but it also locates the PI3 kinase cytoplasmic membrane where its substrate, PI45P2, resides. This PIP3 production then allows recruitment of a family of proteins that have plexure homology domains that specifically bind to this lipid. And we showed in the mid-90s that the AKT family member PH domains bind to this. In later work from Dario Lacey and Lynn Stevens showed that the upstream kinase for PDK1 also, for AKT, called PDK1, also binds to PIP3. And this co-localization of these proteins allows PDK1 to phosphorylate the activation loop of AKT. There's a second TORC2 complex that phosphorylates AKT to fully activate it. And once activated, AKT then phosphorylates a host of substrates. And at last count, there are probably more than 150 substrates of AKT that have been identified in the literature. But these four shown here are highly conserved through evolution and explain many of the things that insulin does, including stimulation of glucose uptake in the muscle through phosphorylation of AS160, stimulation of protein and lipid synthesis by phosphorylation of TSC2, which we showed about 12 years ago or so. Phosphorylation of FOXO regulates, FOXO is a transcription factor, and this can suppress gluconeogenesis in the liver. And glycogen synthase kinase 3 controls glycogen levels. So many of the things that insulin does can be explained by these targets. As long as P10 is there, then you can limit the amount of signaling and ultimately turn the system back off as insulin levels drop, letting the cell reset itself. Now, downstream of this protein TSC2, which we showed as a substrate of AKT back in about 2003, 2004, TSC2 turns out to regulate the TORC1 complex consisting of RAPTOR, MTOR complex, TOR standing for target of rapamycin. So this is the protein kinase that's suppressed by rapamycin, the immune suppressive drug. Work in many species have shown that this complex controls cell growth. Flies, worms, even in yeast, this pathway is controlling cell growth. So in addition to being regulated by growth factor receptors, not only through the PI3 kinase pathway, but also through the MAP kinase pathway, which can redundantly phosphorylate TSC2. This is also controlled by amino acid availability through some G proteins called the RAG complex, RAG-BD or RAG-AC. These sense the availability of essential amino acids in the cell. And unless their essential amino acids are available in sufficient quantities, then it's impossible to activate mTOR even though TSC2 is phosphorylated. Additional important input into this complex is a sensor of energy stress. And we heard a little bit about this last night, in that if cells have low levels of ATP or high levels of AMP, this activates an AMP-dependent protein kinase. It requires an upstream kinase called LKB1, which we showed several years ago, directly phosphorylates AMP kinase and stabilizes the phosphorylated state. And this activation then suppresses mTOR signaling by phosphorylation of TSC2 and also by phosphorylation of RAPTOR and by other mechanisms as well. And so as a consequence, if the cell is under energy stress, it will not attempt to grow, which is an energy-consuming process. Now, one can artificially activate AMP kinase with the drugs Metformin and Fenformin, which cause oxidative phosphorylation in the mitochondria and thereby lower ATP levels and thereby turn on this pathway. And there are many other things that AMP kinase does, as you heard last night, that can help restore energy balance in the cell, including phosphorylation of the TX-NIP protein in muscle to eliminate metabolism of glucose transporters and thereby turn up glucose uptake. Now, if you think about this, this system evolved and is highly conserved through evolution because it makes an important decision for the cell. It's essentially like a computer AND gate that says only if there's sufficient ATP and all the essential amino acids in a cell, and there's also a growth factor telling the cell to grow, will a cell decide to grow. If any one of these events is insufficient, then the cell will stop growing, and this ensures homeostasis in cell growth. We also learned last night that if you have mosaicisms and activation of PIK3CA, the very same mutations that you see in cancer can occur sporadically during embryonic development. And any cell downstream of a cell that's mutated to have a considerably active PIK3CA, PI3 kinase, has a growth advantage around surrounding cells. It can take up glucose and other metabolites at a much higher rate and can grow at a much higher rate. And as a consequence, one sees this localized hypertrophy. It can sometimes be a very late event in embryonic development, resulting in a very small localized hypertrophy, and sometimes much earlier, as we saw a number of pictures last night. And Dr. Sampler and others have contributed to this research. So what's interesting about this network that I showed you earlier is that everything in red in this network is an oncogene and everything in blue is a tumor suppressor gene. And in fact, this is arguably the most, in fact, I don't think even arguably, without doubt, this is the most mutated network in all of cancer. If we look at PIK3CA alone, it is arguably the most mutated gene in all of cancer, particularly in women's cancers where, for example, in uterine cancer, more than 55% of the patients tumors have either point mutations or occasionally amplifications or both in PIK3CA. Cervical cancers very often have amplifications, sometimes mutations. Breast cancers, about 35% have mutations. Ovarian cancers have amplifications and occasionally mutations. But you can see many other cancers also have mutations. And if you look at all the nodes in that network and where you see either loss of function of tumor suppressors or gain of functions of oncogenes that control this growth pathway, then virtually all the cancers that cause deaths in the Western world are driven by mutations in this network. So over 70% of prostate cancers, breast cancers, ovarian, cervical, lung, colorectal, and uterine have at least one mutation, if not multiple mutations in this network. And as a consequence, pharmaceutical companies are very busily trying to develop drugs against every druggable node in the network. And fortunately, most of these are either protein tyrosine kinases, serine threonine kinases, or lipid kinases, and therefore ATP analogs that go to the ATP pocket can be effective inhibitors of this pathway. There are more than 20 PI3 kinase inhibitors currently in clinical trials, and the first drug to be approved was just approved last July, idelalisib, which targets the PI3 kinase 110 delta. Now, Otto Warburg was a German scientist who got the Nobel Prize for his work on mitochondrial metabolism, but he's more well-known today and even in the latter part of the 20th century for what's called the Warburg Effect, his observation that if you take a rat tumor and measure glucose uptake and metabolism, it's taking up glucose 50 to 100-fold faster rate than the surrounding normal tissue from which it evolved. And it's also using glycolysis to metabolize the glucose more so than oxidative phosphorylation. So this is often called the Warburg Effect, and we use it in the clinic as a way of determining where cancers are, in that you can use a radioactive form of glucose, and the cancer typically takes up glucose much better than the normal tissue, allowing you to visualize by PET imaging the location of the tumor. So we asked a few years ago whether we could verify that tumors that have mutations in PIK3CA that PI3 kinase is actually driving glucose uptake into the tumor. And this is an example of this, actually our very first evidence of this, in which we introduced into a mouse a doxycycline-inducible form of the human PIK3CA gene with an H1047R mutation, the same mutation you see very frequently in breast cancer and occasionally in lung cancer, although lung cancer, only about 5% of lung cancers have this mutation. They more often have RAS mutations. And we then asked whether, first of all, did we get a tumor when we expressed this gene? And the answer is yes, we did get tumors after about 4 months of doxycycline induction of this gene. And this tumor looked like a lung adenocarcinoma by pathology. And it has a very strong FDG PET signal, which you can see by this bright spot here. We then asked what happens if we either take the mouse off doxycycline to turn off the gene, or if we gave it a PI3 kinase inhibitor, and in either case, the glucose uptake in the tumor went away within a couple of days. The tumor was still there at a couple of days, but by 4 to 5 days, you could see a pretty dramatic shrinkage of the tumor, and by a week or so, the tumor had completely disappeared. So this indicated that, yes, PI3 kinase is responsible for the Warburg effect, at least in genes that have PIK3CA mutations, and that it's required to maintain not only glucose uptake, but survival of the tumor. Now, interestingly, in humans, there are actually 4 different PI3 kinase genes that can produce this triply phosphorylated lipid, PIP3. There's PIK3CA, CB, CD, and all 3 of these couple to the regulatory subunit P85. And then there's also PIK3CG, which has a different type of regulatory subunit. These turn out to be regulated in different tissues and by different growth factors and hormones. For example, G-protein coupled receptors are the major activators of PIK3CG and also PIK3CB, although PIK3CB can also be affected by other growth factors, in fact require both a growth factor and a GPCR signal to be activated. PIK3CD is unique to hematopoietic cells and is activated by T and B cell receptors, and therefore is necessary for the growth at least of B cell lineage cells as well as B cell lineage tumors, as we showed from knockouts back in the 90s. But it's PIK3CA, as I said earlier, that's really the insulin responsive pathway, and that's also the pathway that's mutated in cancer. And we don't fully understand why this is the only one of these that's mutated, but it's very clear as genes are being sequenced that's the case. Now, what that mutation does is actually enhance the ability of insulin to activate PI3 kinase. That mutation itself is not activating. It only allows the enzyme to be more readily activated by insulin and IGF-1 and some other growth factors. So drugs are being developed against PI3 kinases. In fact, all of these PI3 kinases currently have drugs in clinical trials. The one that was first approved, as I indicated earlier, is the delta inhibitor for treating chronic lymphocytic leukemia. This is a very effective new therapy for this disease. There are also alpha-specific inhibitors, BYL719, for example, being developed by Novartis. And there are also PAN inhibitors for, again, this other Novartis drug, BKM120. And I'll tell you a little bit about some clinical trials that I've had the privilege to be involved with through a team that I put together, funded by Stand Up to Cancer, to evaluate how to use drug combinations with PI3 kinase for treating women's cancers. So the one obvious effect that we knew going into these clinical trials is that if you inhibit PIK3CA product, P110 alpha, with drugs like BYL719 or pan-PI3 kinase inhibitors, that should give insulin resistance because it's going to turn off everything insulin does in muscle and liver, and that's clearly what happens with the drugs that reach a therapeutic dose. They turn off PI3 kinase not only in the tumor but also in insulin, in the liver and muscle and fat, and the patients get acute and sometimes very major insulin resistance, hyperglycemia, which goes away as soon as the drug is cleared. So one question we wanted to ask early on in our clinical trials is whether we could predict based on a change in FDG PET whether or not a patient is likely to have a clinical response to a PI3 kinase inhibitor. So this is an example of a patient that has a PIK3CA mutation. This is an ER positive, PR positive, and HER2 positive breast cancer patient who also turned out to have a PIK3CA mutation. In fact, this is the same mutation that you see, the H1047R mutation, the same mutation that we engineered into the mouse that I showed you earlier. And like the mouse, this patient's tumor has a dramatic increase in glucose uptake, which you can see by these black marks in the liver. because this patient has metastasis to the liver. The patient was given a month of BYL719, the alpha-specific PI3 kinase inhibitor, and at the end of that month, a repeat FDG PET was conducted, and you can see that the glucose uptake into the liver mets has completely disappeared in this patient. The dark spots in the background here are the kidneys, which are clearing the glucose in the bladder, and of course the brain, which takes up glucose at a very high rate. So those are normal patients. So this patient had a dramatic FDG PET response and also had a dramatic shrinkage of her tumor for more than a year. We did a small window of opportunity trial in which we evaluated whether this was going to be in general true, that if a patient shows a decline in FDG PET, in this case in only 2 weeks rather than a month, whether that would predict a clinical response. And in this case, the vertical axis here indicates the FDG PET signal, and everything below the line indicates a decline in FDG PET, everything above an increase. And in blue are the patients who had clinical responses, and all of them had 35 to 40% reduction in the FDG PET signal within 2 weeks. The yellow ones had a smaller reduction or no change in FDG PET, and they had stable disease. And the one patient who had an increase in FDG PET actually had a progressive disease while on therapy. So this correlation has been very nice, but we did not statistically design this to prove this concept, but it certainly is worth further investigation. And if one can get off of an PI3 kinase inhibitor very early due to evidence that you're not seeing a response in glucose uptake, this would allow the patients to move on to other therapies that might be more curative. So I want to then spend the last few minutes on the connection between obesity, insulin resistance, PI3 kinase, and cancer. And the question is, why is there a strong correlation? I think we now know from retrospective studies, multiple independent studies, that there's a strong correlation between obesity, type 2 diabetes, and death from both incidence and death from certain types of cancers, but not all cancers. And so what might this connection be? And for me, the striking evidence that really made me think about this was what I showed you earlier about these women's cancers that have very high rates of mutations in PIK3CA, indicated in the blue bars here, particularly in endometrial cancer and breast cancer. And these are the two cancers that also show the highest incidence of increased risk with diabetes and obesity. So this, to me, looked rather striking. Colorectal cancers also have a high risk and high rates of mutation, as does bladder cancer. But on the other hand, lung cancer, which rarely has PIK3CA mutations, really doesn't show an increased risk at all. So this correlation is really quite striking and is suggestive that there's something about the predisposition to cancer and the ability to activate PI3 kinase, and particularly by the PIK3CA gene, which is the insulin-regulated form of PI3 kinase. And that raises the possibility, which other people have been thinking about for other reasons, that it may not be really obesity or type 2 diabetes that's causing cancer, but rather the intermediate stage of insulin resistance. Because at that stage, serum insulin levels are quite high by definition. And also free IGF-1 can be quite high in this disease as well due to low levels of IGF-1 binding protein. So we would argue then that this high level of insulin can activate PI3 kinase in any tissue that has insulin receptor in it. And a number of epithelial tissues do. And the tissues that have the highest level, besides muscle, liver, and fat, are endometrial tissue and breast tissue. And what's more, as we look at tumors that evolve in those tissues, very often the tumor has 10 to 20 times as much insulin receptor as the normal breast or endometrial tissue. So this major upregulation of the insulin receptor allows that tissue to take advantage of the high serum insulin availability to drive PI3 kinase. The mutations in PI3 kinase then allow insulin receptor to even more readily activate PI3 kinase and avoid insulin resistance. And so as the patient's muscle and fat and liver become insulin resistant, the tumor actually becomes more insulin sensitive due to these mutations. So this can drive neoplasia, more redox potential, and more redox damage of DNA, and ultimately result in full-blown cancer. So that's the model, and I think the evidence supporting this is becoming stronger by the day. So just a little bit about basal insulin, a very simple model of insulin signaling where you eat carbohydrate, the glucose enters the bloodstream, that causes pancreas to release insulin, which goes to the liver to suppress gluconeogenesis. I didn't show the muscle here, but there the glucose gets taken up by the muscle, and that brings the glucose back to homeostasis again. And so this simple network really allows very fine-tuned control of serum glucose levels. If the tumor has insulin receptor, or a growing neoplasia has insulin receptor, then this elevation of insulin can help drive glucose uptake into the tumor. If the patient has insulin resistance, then the serum insulin levels can be orders of magnitude higher, and this will allow now this growing neoplasia to have a huge advantage, particularly if it picks up a mutation in PIK3CA. It allows it to respond to insulin even better, and we think this is what's really driving these early tumor growths and can continue to drive tumor growth throughout the disease process. Now, we have ways of trying to keep glucose levels low, and endocrinologists worry a lot about making sure the glucose levels are low. But some of these mechanisms of keeping glucose down actually raise serum insulin levels. Obviously, injecting insulin will raise serum insulin, and agents that stimulate insulin release from the pancreas will raise serum insulin. That will bring glucose in control, but at the same point, serum insulin is going to be even higher, which could be driving these neoplasia. In contrast, if one uses drugs such as metformin or the sodium glucose co-transporter inhibitors, then you can lower glucose without raising insulin, and I would argue this might be a better choice. Metformin, as we showed several years ago, will cause activation of AMP kinase, and that can also help suppress gluconeogenesis without elevating insulin. And as a consequence, one can keep insulin levels low with this drug. So might metformin then be our best cancer drug? Well, there's a whole series of papers that have come out over the last 10 years that have argued this from retrospective studies. So if you take otherwise matched type 2 diabetic patients and ask for those taking metformin versus other therapies, the other therapies typically being insulin or agents that stimulate insulin release from the pancreas, the patients that take metformin have a 25 to 30% reduction in cancer deaths, which arguably makes metformin the most successful anti-cancer drug ever developed. But the mechanism by which it works has been debated. Some have argued that it may get directly into the tumor and activate AMP kinase there and that may be true to suppress tumor growth, and that may be true. But metformin does not get into most tumors nearly as well as it gets into the liver, where it suppresses gluconeogenesis. So I would argue that it's really suppression of gluconeogenesis in the liver that is allowing metformin to maintain insulin levels low and thereby suppress gluconeogenesis. So for this reason, for patients who go on to the clinical trials that we've designed, we have a very firm request, if not a requirement, that the patients be managed on metformin or a sodium glucose co-transporter inhibitor to keep insulin down, because if insulin levels rise to the level that it overcomes suppression of gluconeogenesis, overcomes the suppression of insulin signaling in the liver, then it will probably also overcome insulin signaling in the tumor. And so adding metformin to a PI3 kinase inhibitor is almost certainly going to increase the effectiveness of the drug on the tumor as you manage the glucose levels with metformin. So the final slide then, or figure slide at least, we would argue then that there's a huge risk as in the Western world we become more and more insulin resistant due to eating high glycemic foods, eating too much, exercising too little. All of this contributes to high levels of glucose, but more importantly, I would argue, high levels of insulin, and these high levels of insulin are almost certainly helping to drive tumors. And just in the last 3 or 4 months, a series of papers have come out that very strongly support this model. A paper from Michael Pollack's laboratory back in November showing that only patients with evidence of insulin resistance showed decreased breast cancer growth when treated with metformin. I think this very strongly indicates that it's lowering the insulin level that's causing this advantage with metformin treatment. A second paper from Strickler's laboratory broke down cohorts of patients, both of which were obese, equal in weight, but the subset that had insulin resistance went on to develop breast cancer. The subset that were obese that did not have insulin resistance did not have a higher risk of breast cancer. So I think that really argues it's the insulin resistance, not the obesity, that's causing this disease. And then finally, a very nice paper by Stambloch's laboratory, in which in a neoadjuvant setting, they looked at patients prior to metformin treatment and after metformin treatment through biopsy and then surgery to evaluate what's going on with insulin signaling in the tumor and PI3 kinase tumor in the tumor. And they found that metformin reduced the tyrosine phosphorylation of the insulin receptor and reduced AKT activation in the tumor in patients that were insulin resistant. So this, I think, overwhelmingly argues for the model that I proposed. And I'll stop at that time because I think I'm running out of time and just summarize the major findings that I discussed today. PIK3CA gene product mediates almost everything that insulin does. PIK3CA is one of the most mutated oncogenes in cancer. The oncogenic mutations in PIK3CA are not themselves activating, they just allow insulin to activate PI3 kinase better. Most solid tumors express high levels of insulin receptors, particularly breast, endometrial, and prostate, and have sustained high serum insulin. As a consequence, sustained high serum insulin is almost certainly driving the growth of those tumors. Retrospective analysis has shown that type 2 diabetic patients on metformin have reduced cancer deaths compared to matched patients on other therapies, and the benefit of metformin and other therapies that lower serum insulin, including diet and exercise, is causing lowering cancer deaths may be a consequence. I don't think we can say this conclusively yet, but the data is becoming overwhelming that this is a consequence of reducing insulin-dependent tumor growth. And finally, insulin resistance may be more dangerous than obesity or type 2 diabetes with regard to cancer risk because of high serum insulin that goes untreated. And I'll stop there and thank you very much.